we're definitely stuck in kind of a sick care mentality rather than being more proactive about fixing things before they're broken, right? And there's things like that you can get away with in cars and houses, but there's things that you can't, right? So in timing belts on cars, you have to replace them before they break, right? And that's just normal maintenance. You got to change the oil, before your engine seizes up, or going to be $30 or something to change the oil in your car, whereas it's going to cost you like 5 or 10 grand to rebuild your engine if you don't change your oil. That's the kind of mentality that I think we need to shift to in medicine.
Hi, everyone. Welcome to episode 14. I think it's going to be 14 of the Pauling Principle. I'm here today with Matthew O'Connor, Oki. And I'm actually, very excited to have this conversation. We haven't spoken in probably, 6 months, a year. Like, when was the last time that we chatted? Like, I think, maybe in one of the LBF events or maybe the ARDD last year. It's been a while.
Yeah, it might have been a year. We traded a message or two here and there, but yeah, it's been a while, so it's great to catch up, Javier.
So yeah, maybe to get started, why don't you tell us a little bit about, your path, to, longevity, if, what kind of, uh, uh, I don't know, like, help you, you started thinking about it, was it, from a very young age, or, did it come later? And then, what led you to you founded in, I guess, Cyclarity now, Underdog, Therapeutics, before, and, you kind of, your path in life.
Sure. So, I did start thinking about this from a pretty young age. Since I was 16 years old, pretty much the only thing that I've wanted to do is work on aging and longevity, and so I set out to go and get a PhD and then do research on the biology of aging with a goal of doing something to fix it, because I've thought for over 30 years now that it's the world's biggest problem, and my opinion hasn't changed. So I'm still working on it.
Yeah, I mean, maybe not a, I don't know, super happy thought, but this is one of the things that really drives me crazy, right? Like, how some people don't understand I don't know, the significance of it, right? Like, maybe they just don't think there's something that can be done, or like maybe they just, accept, fate. I don't know, I don't want to go, down too deep down the rabbit hole, but why do you think, there's not more effort in longevity, or like more money being spent? Like, there's definitely a lot of money, a lot of resources, a lot of, mind share, spent on, curing cancer, right? Like, so why is it not the same for delaying aging, for example, or fixing?
In some ways, it seems intuitive to me that that things that seem inevitable are things that people don't, have some resistance to, right? So, people think war is inevitable. So why bother, trying to work for peace? People used to think that, childhood, that maternal mortality was totally normal, right? That childhood mortality was totally normal. That starvation was totally normal. Why bother trying to feed the world? And now, like, we're closing in on basically being able to solve all of these things. Certainly, war is much more present in mind today because we can see it on TV so much, but actually, a fatality rate, a violent fatality rate, keeps dropping on average, decade after decade, things like that. So I think, you know, infectious disease, I think, is a good example that seemed like it was really normal until it wasn't, until we had, first, we had antibiotics and vaccines, and now, dying of infectious disease just is like a huge outlier. It's pretty unusual, right? So now, after that, then people say, oh, HIV, it's an incurable virus, but people say, what? We've cured all these other diseases. Why can't we just cure that? So I think longevity just hasn't crossed that threshold yet. I think it will, and we'll like some number of decades from now, we'll look back on today's attitudes, just like we look back on those other attitudes that I was just talking about, and say, why were these people so naive? Why did they think that they just had to die of old age? But we're not there yet, and so, I think we'll hit that inflection point, in the not too distant future, but we just haven't crossed it yet.
Yeah. So many interesting things. I actually, now that you mention it, I remember a while back doing this exercise with ChatGPT or Claude, saying, "Well, what are the costs of death of humanity across history, right? Across millions of years, I guess, of Homo sapiens." And I think the estimate that it came was like maybe 20-30% of people actually died because of malnutrition, and then another 40-50% people probably died because of infectious disease of one type or another, right? And then there's everything else, trauma, like violent conflict and old age, right? Um, and what you said is very traumatic, right? It's very hard for us to imagine today, right? That people would die of malnutrition or infectious disease, right? But back in the day, that was normal, right? Like the vast majority of people would die, either at childbirth or when they cut themselves, through one way or another, and most people would never reach, 40, 50 years of age, right? I think like up until what the 17th century, the average lifespan was like 35, 40 years, something like that, which is crazy, right? Like how different the world is. And now like, we see people in their 60s, 70s, 80s, 90s, 100, right? And like we don't think twice about it.
Yeah, yeah. You look at, kind of good problems that we have now with, retirement plans and such, having challenges because when they were created, when Social Security was created, our average lifespan was in the 40s, and now it's like 80. So it's amazing progress when you look at it over a long period of time, even though it's frustrating to work on stuff, day by day, year by year, and have a hard time seeing the difference that it makes. But we've come a long way, and even in the longevity field, which has only existed, depending on when you count it, for 2 or 3 decades, it changed so much since I've been in it.
Um, who else should join the field? Now that you mentioned you, so you decided, early enough that you wanted to PhD. You started, I guess, at the Back Institute, was it? And
So I got my PhD at Baylor College of Medicine in Houston, working on telomeres on cell senescence, and then I went to UC Berkeley, and I worked on muscle aging, muscle stem cell aging, and then I went to join the SENS Research Foundation, which was just getting started then in 2010, and I helped build the lab that we made there in Mountain View, the research center that they're still in. I was there for 9 years and became vice president of research. Running, overseeing the all the research that we're doing in the lab there, and also the extramural research that we were funding around the world, mostly in academic labs through grants. And then I discovered the technology that was the basis of Cyclarity, and spun that out in end of 2019, and that's what I've been doing since then. So that's my career path in a nutshell.
awesome. So you- like, what is the I guess the technology behind psychedelic? Well, of course, we've talked about this before, right? But maybe for the benefit of somebody that is uh entirely new to this, who doesn't understand much about cholesterol or cardiovascular disease or cyclodextrins or whatever, right? Like, what is the basis of what you're trying to accomplish in your company?
Yeah, so we invented a technology using a uh, a kind of molecule called a cyclodextrin, which is a cyclic carbohydrate that has all kinds of interesting uses and applications. They've been in science broadly for about 100 years, and they've been in medicine for about the last 25 years. Most of what they've been used for in medicine is to deliver drugs. So let's say you have a chemotherapeutic drug that you need to inject into somebody's veins, but it's not soluble in blood or in water, since blood is based on water, and so you need to get it into a water solution before it can, get in the bloodstream. So you mix it with a lot of one of these kind of generic, cheap, safe cyclodextrins, and it goes into solution, it mixes in, and then you can deliver it safely into the bloodstream. So that's used in dozens, if not hundreds, of drug products now. And so then in 2015, there was the first drug based on this technology was approved. It's called Bridion. It's owned by Merck, and it wasn't invented by Merck. Uh, it was developed by smaller labs like mine, but it was a this unlike those other cyclodextrins that I was talking about that are kind of cheap and generic and, not very specific for their job, but kind of non-specific that you can use them, for almost anything. This had one very specific job, which was to grab onto a paralytic that's commonly used in surgery, and to pull it out of the bloodstream so that the patient will be released from the paralysis like right away, and then they just pee out the paralytic and the reversal agent. So it's an antidote against a paralytic that you can just get rid of, and that's very very specific. So it will only grab onto it, and it was designed to do that one job. And get rid of it. So that was a lot of the inspiration for the technology that we developed. So we built on top of that kind of idea where we're sticking two of these cone-shaped cyclodextrins together in a certain configuration so that we can wrap around a target and bind it really, really tightly. So we have this dimerization technology where we stick two of these together, and it kind of can grab onto a target that it's been engineered to be very specific for, like Bridian, and grab it with very high affinity and high specificity, and float away and operate on the same mechanism. The first target that we chose for that was oxidized cholesterol, which is kind of you've heard of the good cholesterol and the bad cholesterol. I think of it as the really bad cholesterol because unlike the supposed bad cholesterol, which is LDL, low density lipoprotein cholesterol, you actually need LDL cholesterol to survive. It's actually the delivery cargo for cholesterol in your body. Every cell in your body needs cholesterol, and LDL ships it around. The only problem with it is if it gets too high, then it ships around too much cholesterol and too much absorbs, especially into your blood vessel walls, and that can clog up your blood vessels. But I wanted to pick a target that was just bad. That if we could get rid of all of it, you'd just be better off. And I think that those are the ultimate drug targets. And so we picked this really bad oxidized cholesterol that's just toxic, and built a drug to grab it, pull it out, and excrete it. And that's what we've built, and that we're testing now.
Okay. Lots of interesting things to ask about. So, um, why why are cyclodextrins like so safe or safer, right? Like, I imagine like I'm kind of trying to picture it in my head, right? Like, like of course you have like I don't know glucose or fructose or all sorts of other like sugars, right? Or like I guess glucagon, that accumulates in muscles or whatever. So like it's a cyclodextrin, just like a chain of little sugar molecules that is kind of form into a cycle or something like that.
Yeah, it's exactly, so there's 3 different sizes of cyclodextrin, 6 or 7 or 8, glucose molecules stuck together, but they're stuck together in a circular cone, so they actually don't get, they're more like a fiber than a sugar, so you can't digest them and get energy out of them, and they don't, unlike glucose, which is a pretty relatively highly reactive molecule for something that's just floating around everywhere. Glucose can, it's actually part of the aging process. It can bump into things and react with proteins and damage them and cause stiffening in like blood vessel walls, and it can cause damage to peripheral nerves and cause nerve damage in diabetes. But these cyclodextrins actually are very non-reactive because everything's sort of wrapped up in a cone, and it doesn't react enzymatically or non-enzymatically with anything in your body, and it doesn't interact with your immune system. So they tend to stay in our bodies whether you eat them or they're injected. They stay for a very short time, and they'll just get cleared right away. You just excrete them whole. So you excrete them, unlike most drugs, which have a pretty long and complicated metabolic path to clearance. Cyclodextrins, the way that you take them in is the way that you like, get rid of them, and they don't react with anything. They don't change. It makes them pretty easy to study in terms of safety, and and so that's what makes them so attractive in terms of safety, is how non-reactive they are.
Mhm. Interesting. And I guess what is maybe original in your therapy, right? It's like this thing tries to find one of these toxic, uh, substances or a target that you want to remove from the bloodstream. But normally people would do this with an antibody, right? Like you, I guess, find or design an antibody, and then you use that antibody to get the immune system to clear the thing. So, like, what would be the benefit of using a cyclodextrin versus an antibody? Or, like, could you use one or the other? Are they interchangeable or not? Like, what kind of problems may come up, if you use an antibody, I guess.
Yeah, I think it's a really good question, and I think it's a big opportunity for the field. I like to think of cyclodextrins as being sort of halfway between uh, small molecule drugs, which is the class of drugs that most people think of. Whether you take an aspirin or something like that off the shelf, those are all small molecule drugs. Anything that you're taking orally, most chemotherapeutic stuff like that are all small molecule drugs. Now, antibodies are really really different, and antibodies you actually generally you don't you can't program them, and that's sort of changing now. But traditionally, you sort of create antibodies with by challenging an immune system with a antigen, right? So you want to get rid of something, like something like, let's say, a virus particle, right? So you challenge with it, and then you raise an antibody against it, and then it can grab onto it. The nice thing about antibodies is they're like super super tight binders, and they can be really specific, meaning very picky for what they're grabbing onto. Ours is completely engineered, so it's not made through this kind of natural selection process that the way antibodies are kind of grown. Um, we design them computationally, we test them computationally, and then we synthesize them and test them at the bench, stuff like that. But we build them synthetically. So they're built on the basis of these basic building blocks, these cyclodextrin building blocks that are natural compounds. They're made by microbes and grown in large vats. But then we do synthetic chemistry on top of them, and we build in all of that specificity for the target. So in a way, they're sequestering the way that antibodies are sequestering, like you were saying, but we're building them more like synthetic molecules, like like traditional rational drug design. So it's kind of halfway. I think of them as sort of halfway between antibodies and small molecule drugs, and those are the two biggest classes of drugs in the industry. Um, and so I think there's a lot of opportunity since there's only, there's hundreds of both of those classes of drugs, or a lot more small molecule drugs. But in terms of market size, the antibodies are even more than the small molecule drugs. But I think there's a huge opportunity for something that's kind of halfway in between those two, and that's how I think of the cyclodextrin. So they behave kind of like antibodies. They're made in a different way. Antibodies interact with the immune system, so a big fear whenever you make a new antibody is, what is it doing to the immune system? Is the immune system reacting to it? And people are always working on iterating antibody technology to try to make it less immune reactive and only to do its job of grabbing onto something. But it's always an issue anytime that you're injecting a protein into the bloodstream. You have to worry about an immune reaction, like an allergic reaction, or another immune reaction to it. You don't have to worry about that really at all with cyclodextrins. You do have to worry about it with antibodies. So that's some of the compare and contrast between antibodies and cyclodextrins and small molecule drugs.
Yeah, yeah. Because I guess the danger with antibodies is if it binds something that it's not supposed to bind to, right? Like it can cause, I guess, a lot of trouble, right? Like if it somehow, um, or if it binds, maybe really next to something, it doesn't even actually have to bind. Like if it binds next to something else, and then it kind of, there's I don't know, I guess a T cell or something like that that find that and decides, oh, this cell is toxic or whatever. That T cell may end up killing something that it shouldn't be killing. I guess like that's how people develop like autoimmune diseases or whatever. It's just immune system just malfunctioning, right? And trying to destroy something that it shouldn't.
Yeah, by analogy, the the specificity for the target is really key with making cyclodextrin-based drugs also. And so when we built our first drug, we trained it to bind the oxidized form of cholesterol, which only differs from regular cholesterol by one oxygen atom. So it was challenging to do that, but what we did is we trained it for the target oxidized cholesterol and against regular cholesterol. And that's what gave us the ability to build something that was specific for the target. And in fact, if we grab too much cholesterol, we can pull it out of cells, and your every cell membrane has cholesterol in it. And if you pull too much cholesterol out of a cell membrane, it will break apart, and that cell will die. So, we have plenty of failed candidates that combine cholesterol very tightly, but they'll just tear cells apart. And so that's how we addressed that problem, was by having like a positive and negative selector, both for our in silico modeling for the computational modeling, and then at the bench testing was to test for the target and against a anti-target.
Mhm. And I guess you're starting with uh, oxidized cholesterol because of the potential to impact cardiovascular disease, right? I guess the biggest cause of death for people today. Uh,
We actually approached that decision from the opposite direction. So I was at a aging research foundation, and as kind of more of a basic scientist, I it was a target that we know accumulates with age everywhere that you have cholesterol, which is everywhere in your body. And so it seems funny to people when I say this now, that I didn't build a cardiovascular disease drug. I built a aging drug and are applying it to cardiovascular disease because that's the best disease of aging that is most dir directly connected between the oxidized cholesterol and the disease. So, yeah, I actually came at it from aging, and then, we thought, what's the best application of it? Well, it is the biggest disease in the world, but we think about it as it's part of the basic aging process that causes that disease, and so I think that this drug will also be effective against many other aspects of disease. Brain aging, brain is where you have the most cholesterol, and so likely to have the most oxidized cholesterol in the brain compared to the rest of your body. But in terms of something that's really obvious that happens with age, it is the plaque that builds up in your arteries. So that's what we're going after first.
Yeah. And that's a very interesting, maybe, uh, viewpoint, right? And so I'd love to go a little bit as a bit deeper into it, right? So I guess, some people have the opinion that, I don't know if there's like a great definition of aging. There's definitely not a great definition of aging that everybody would agree on, right? But like there's some people veer more on the side of aging is caused or mostly caused by accumulation of damage and accumulation of molecules that shouldn't be there. I guess some people think that aging is maybe uh, a process that is somehow predetermined, through different mechanisms, I guess, of epigenetic regulation or other things, right? Do you subscribe to that view that aging being mostly determined by accumulation of damage or accumulation of molecules that shouldn't be there?
I do, and I don't like to get into the debate about what causes aging? Why do we age? What is it about evolution? Is aging programmed? Stuff like that. I'm not sure it matters so much to get into those esoteric academic debates. I think the damage accumulation is a simple explanation, and it's not really the nice thing about it as a model for therapeutic strategy is that it's kind of agnostic about whether aging is programmed or not or anything like that. So regardless of whether or not you can win the argument of which theory of aging is most correct, or that kind of thing, you can look at a piece of damage. You can look at a senescent cell and just think that that's a good target for therapy. You can look at damaged cross-linked proteins and say that's a target for therapy. You can look at damaged oxidized molecules like cholesterol and say those are targets for therapy. Uh, and they're, like I was saying before, that they can be perfect drug targets because if the optimal amount of that in your body is 0, then it's harder to overdose on it than other drugs where you're just trying to get, say you're trying to get your blood sugar down into this perfect range, your fasting blood sugar between, 70 and 100 milligrams per deciliter, right? That's kind of a tightrope walk, and of course it's important to keep your blood sugar and your cholesterol levels within a healthy range, but you're just sort of talking about homeostasis and rates of aging, as opposed to taking one concise piece of aging and getting rid of it, and then just one piece at a time, and then you can fix the biological system that you're working on, and you're not going to have side effects from that if it really is just damage that's accumulating with time, from mostly from entropy.
Mhm. Yeah. Which to me, as an engineer, makes perfect sense, right? Like if you know that there are some processes that you can classify as damage, right, and you can prevent that or revert that damage, right? Then like maybe you can't prove that, the organism, lives longer, right? In a traditional clinical trial, but it seems reasonable, right, that you would cause that effect, right? If you just, go ahead and remove that damage, right? And I think, your example is really good. But I mean, there are many other diseases that are the same way, right? I guess, in some ways, a lot of the effects of like Alzheimer's, for example, are caused by, accumulation of like amyloid beta and tau fibril and so on and so forth, right? Uh, and in many ways, I guess like, senescence of neurons, for example, ends up causing, types of neurodegenerative disease or whatnot, right? So I think, in my mind, whether the damage version of aging is right or not is, I agree, it's irrelevant, right? Like what matters is, can we go ahead and fix it, right? And then if we can fix it, then, we'll probably will live longer.
Yeah, I mean, that's exactly, where I come at it from, and I think that it would be beneficial if we start thinking about, um, treatment of biological systems from that perspective and get a little bit less uh, outcomes, sort of be more in this way, be more process oriented and be less results oriented, and think less of how do we, get something that will make us live longer, or get something that will improve our memories, or improve our heart function, or whatever it is, and think, let's fix, one thing at a time, and and then things will eventually get better. It's not that you don't want things to get better, that's not the ultimate goal and the ultimate reason, but I think if you get too fixated on, well, we got rid of some, amyloid plaque in the brain, but, grandma's memory didn't come back, so it's not working. Uh, well, maybe you needed to get rid of that, amyloid plaque, and you needed to get rid of the tau fibrils. Uh, or maybe you needed to get rid of those 2 and the oxidized cholesterol or something like that. But
Right.
if you get rid of those, one at a time, and just, knock them out, you're going to get better, you're going to feel better, think better, eventually, but we should think about it not just, "Oh, I've got some rotten wood on the, foundation of my house, is my house gonna fall down if I don't replace that rotten wood, or, or not?" Is kind of it's not necessarily the best question. Well, it's rotten wood, like, you should get around to fixing that, and your house is going to be better off. It may or may not like fall down, this weekend, or next year, or in 10 years, but it's good to stay on top of that kind of maintenance.
Yeah. And that's a great analogy. I think the first time that I heard it was probably uh already great talking about cars or maintaining cars, right? But maybe a house is a better analogy, right? I guess sometimes in modern medicine you go and say, well, if the roof breaks down, then you have a big hole in the roof, then you go ahead and try to fix it, right? Like that's what it means, going to the doctor and then telling the doctor, hey, I had a heart attack, what do I do now, right? Whereas if you, every year you go ahead and, clean the roof, and every once in a while, kind of, clean the gutter and like, replace a few tiles here and there, right? Like that roof, there's no reason why it wouldn't last for, 100 years, right? Um, and I guess, I mean, it's one of the unfortunate things in modern medicine where like, people are not necessarily as incentivized to go and do that maintenance as opposed to just tackle, when the roof breaks down entirely.
Yeah, I mean, we're definitely stuck in kind of a sick care mentality rather than being more proactive about fixing things before they're broken, right? And there's things like that that you can get away with in cars and houses, but there's things that you can't, right? So in timing belts on cars, you have to replace them before they break, right? And that's just normal maintenance. You got to change the oil, before your engine seizes up, or it's going to be 30 bucks or something to change the oil in your car, whereas it's going to cost you like 5 or 10 grand to rebuild your engine, if you don't change your oil. That's the kind of mentality that I think we need to shift to in medicine.
Yeah. Maybe shifting gears a little bit. how so like you knew about this target and cyclodextrins, there was inspiration of the other one, but like what was the process that you found to go through that? Like, how did you go ahead and start, designing this molecule? Like what kind of, there's not like a Google for molecules that you can go and say, oh, let me, search for a molecule that does this, right? Or I don't know, like a 3D designer tool that you go and start, moving atoms, and then it tells you, oh yeah, this works or doesn't work, right? Like, so how does that process look like?
Yeah, so, we built, tools, or, maybe more accurately would be that we hacked existing tools, to build our computational chemistry platform that we use. So we kind of the traditional application, or the most common, most traditional application, is that you have receptors, say on the surface of cells, and then you have ligands, proteins, mostly, or whatever kind of ligand is going to get bound by that receptor, and that's a very traditional application for molecular simulations, which you know a lot better than I do. We decided to take that and pretend that the cyclodextrin was like a receptor, and that our target, a form of cholesterol in this case, was the ligand. And then we changed the parameters of the software to to optimize for away from proteins and towards uh, on the uh, on the receptor side, which is now the cyclodextrin, to optimize for those. And then the ligands, we tried not to mess with that too much because we like to be able to bind anything that could fit inside the cavity of the cyclodextrin. So we optimized that. We worked on that. We still work on it now. We're always constantly tweaking it to trying to validate our computational predictions with real world examples, and make it better. But that's simply what we did.
And I think you mentioned also, it's another thing that I was writing in my notes as you was talking about, that you were talking about, that cyclodextrins are, I guess they're not metabolized, but they are excreted very quickly, which I guess is an advantage, right? Because the less time a drug spends in the system, the less chance it has to, you know, I guess mess up something else or whatnot, right? Like, makes it safer.
Yeah, well, a lot of people see it like traditional pharma people see it, and they think that that's a problem, that that's a mistake. They say, why don't you design it to stay in the system longer so that it can act longer? Uh, our drug will stay in the system for a couple of hours. We're testing it now in humans. We know how it behaves in animals, and we expect that it's going to be a couple of hours in humans. Uh, uh, but we when we tested it in the lab, it actually does its job within about 15 minutes. So most drugs need to act over a very long period of time. Say you have a drug that is going to block pain, uh, right? You want it to do that for a long, long time, and so you might design your drug to last for hours or even days, or maybe it accidentally lasts for days, and you say, "Well, that's okay. They'll just, not have pain longer." But then you have all these complications of things, that will last for their half life might be as long as several days. But then, if you take it, multiple days in a row or something like that, it can be in your system for, weeks afterwards. And it becomes sort of chaotic to predict what kind of effects that's having long term on people's bodies, right? So, yeah, we designed our drug to to last for a very short amount of time, but do its job even faster than that and just be done. So the simplicity of the mechanism of our drug is that it doesn't need to act multiple times, or have a very lasting um, presence to do its job. It needs to grab one molecule of the target and then float away. Once it's grabbed and it's holding on to that one molecule, its job is done, and you need to pee it out. That's it's gone, and you're going to need more of the drug to get more of the target gone until most or all of the target is gone, and then the job is done. So it's a bit different in that way from traditional drugs, which are acting on something in your body, like a pain receptor or something like that, it needs to act repeatedly on, or it needs to signal for a cell signaling pathway, and you need that to amplify. And so you have one molecule that amplifies the signaling pathway, and you need that to last for a certain amount of time. Our drug isn't doing any kind of signaling; it's just grabbing one thing and going away.
Yep, yep. Of course, the drugs that bind to pain receptors, they're likely to, lead to overdoses as well, right? And all sorts of, side effects, right?
Yeah, so yeah, if it's still there the next time somebody takes more of the drug, then then they just have a lot more in their system than they did when they first took it, right? So it gets messy, especially when you're allowing patients to take something on their own, or whatever it is, whether it's a street drug or even a prescription drug. If they're taking it on their own and they can't measure the effects or something like that, they don't have somebody looking over their shoulder telling them to stop or something like that. You're right. Then they can get addicted, and they feel less of an effect, so they take more, and they're taking it frequently while they still have the drug in their system. Um, yeah, we're trying to avoid that
Yeah.
kind of scenario.
What was it? The therapeutic index, right? Is that what therapeutic index is, right? Like, the rate of drug that could cause toxicity, divided by the rate that has the therapeutic effect, right? And, you want that to be as high as possible.
Yeah, yeah. Um, yeah. So we have a very wide index with our drug, and in fact, it's almost impossible to kill an animal with our drug. We were able to achieve it in a small number of animals with an extraordinarily high amount of our drug, and we think, like I said before, that it's from stripping cholesterol out
Mhm.
of cells. But we were actually not even able to determine a final cause of death in like the rare animal that we could actually kill with an super overdose of our drug, which an overdose level that would be physically extraordinarily challenging for a human to accomplish. Like to actually deliver like that much into them. It's not the kind of thing like some drugs that are really, really potent, that you could accidentally take, 10 times or 100 times too much if it's mixed incorrectly. Our drug is the way that it's delivered, you would have to physically deliver such a high volume of it that it would be completely obvious that you were taking way too much.
Yeah. Like literally open a pill, bottle and just like eat every single pill and, many, many bottles, I guess, right? Of the drug. Yeah.
Yeah.
Um, And all this was interesting, but like, 'cause I was just reading earlier this week about a drug that was just approved for HIV. It was a small molecule that supposedly has a half life in the order of months, which is pretty much unheard of, right, for a small molecule. So that was interesting. I guess in some cases you do want your half-life to be literally as long as possible, right? Like this is basically an antiviral, right? Like it's a small molecule that kind of, prevents something happening in the capsid of the HIV virus first to prevent the virus from working. Uh and I guess if you have a drug that just, clears out some, you know, toxin from your system, for example, right? Like, the fact that, it stays in your body for 2 hours and then goes away is just not a problem, right? Like if you need to take it again tomorrow, then you take it again tomorrow, right?
Yeah, you take it as many times as you need to take it, and then you go away for a year or 5 years or 10 years and take it again, because this stuff takes decades for it to build up, which is really the opposite scenario uh, from HIV, which is a permanent chronic disease. So, yeah, I mean, that's a perfect scenario where you do want the drug to last for as long as possible, and so if you can get a half-life of months, then that is a good thing for that scenario. Obviously, the perfect damage repair approach would be to get rid of the HIV infection, but that that's proved to be really, really challenging, and we'll solve that problem someday, but hasn't been solved yet. So you have to take these, sort of infinite treatments for HIV. And there's a lot of chronic conditions, usually non-infectious conditions, but there's a lot of chronic conditions where you just have to take a drug every day, or if you can get something that will last for months, then you only have to take it once every few months. So there's getting more examples of drugs like that. There's a cholesterol lowering drug that they did something similar with that has a half-life of a month or 2, and you take it once every 6 months, and it keeps your, LDL cholesterol levels low, all the time.
Yeah. Um, this is actually one of those things that I find fascinating. I don't know why there's no more research on it, right? But like I suspect there are many infectious diseases that have a very drastic impact on aging. Right? Like one of the papers I remember reading a while back that made a lot of sense to me was there was this study that they did. I think it was in Taiwan, where they were able to draw causal conclusions between having one of the herpes virus infections and developing Alzheimer's disease. Like people that and that was because they at some point they started vaccinating everybody, right? So you had, two very clear, cohorts, people that were, born before this date, they were not vaccinated, people that were after that date, they were vaccinated. And then they realized, if you were vaccinated, the odds of developing Alzheimer's thing was like 20-30% lower, something, very, very significant. And I think the kind of, rationale for that was like, well, maybe the kind of, the viral infection, even though it doesn't have a lot of, for most people is asymptomatic or whatever, or like, it ends up causing, I guess, inflammation, right? Or, or other things, right? And then, if you can just, get rid of that infection altogether, you kind of, reduce the risk of developing Alzheimer's, which means, probably you would end up living longer, right? And in my mind, it just makes so much sense too, right? Like if you can solve the problem entirely by avoiding the infection, you kind of, solve one of these things, right? If you solve enough of them, eventually you will live longer, right?
I totally agree. I think this is a really understudied area of kind of at the interface between infectious disease and aging that is is really underappreciated. And I know I've read some of those same studies. I think that you're talking about that. Herpes one, which is herpes two, is the what more commonly known as as a sexually transmitted disease. That's really annoying because you get lesions on your genitalia and stuff like that. But herpes one is considered, harmless, and whatever, like 90% of the population has it. And every once in a while it causes cold sores in some people who have it. But, it's not killing people or, drastically impacting their quality of life. So, it's pretty much harmless. But, over the course of decades, actually, it's probably not harmless. It's probably damaging your brain and contributing to dementia. And so, these persistent viruses like the herpes viruses and, CMV in the immune system is a big one. I think it's like 80% of people who have that. Uh, and it seems to really mess with not your ability of your immune system to function when you're young, but when you're old, it turns out that you have, some huge percentage of your immune system that's trying to fight this infection that's basically harmless, except that, it's wasting all the energy of your immune system, and now your immune system doesn't do as good a job of fighting off, the flu, influenza or pneumonia or something like that. So,
And you don't want it to be active all the time, right? Because
exactly.
the more active it is, the more chance it will actually end up messing something that it should, right? Yeah.
Exactly, exactly. Contributes to inflammation, chronic inflammation, that kind of thing. So, yeah, I think that we really need to think longer term and, less, yeah, oh, well, the virus isn't, killing people today, or there's finally some awareness about HPV and cancer, and so we have vaccines for that now, but I really like to see more activity on, not just obsessing over, COVID and the flu vaccines, which can kill you in one shot, but, these chronic diseases, both for vaccines for prevention, but also, how can we get rid of chronic diseases? Everybody agrees that we should get rid of HIV, but we should also be thinking about some of these, maybe some of these other ones will be easier to get rid of than HIV if we put a fraction of the effort into curing them that we do into trying to cure HIV. Maybe it would be relatively easy to get rid of CMV if anybody actually bothered to do it, but I've never seen anybody really seriously work on that.
Yeah, yeah. No, 100%. Of course, you're, preaching to the choir, right? As they say, right? I completely agree with that. I was actually, just recently doing a little bit of reading about that, about a different one, which is Toxoplasma gondii, T. gondii. I don't know if you've heard of it, right? Like it's a parasite, right? That typically infects cats, but it can infect, I think, people as well. It's a protozoa, and supposedly it has a big impact on fertility. For example, I was thinking, well, I mean, like we've seen, maybe kind of, on a society level, we've seen, fertility drop drastically, right? Over the past few decades, right? And I wondered if some of it is actually caused by, infectious disease, for example. Um, Nobody seems to be doing any research on it. And I'm thinking, well, I mean, like, people will go and spend, tens of thousands of dollars on fertility clinics or other things, right? But there's basically zero research being done about, some other, things that may actually be, um, impacting fertility. Anyway, it's a little bit far away, I guess, from longevity, right? But like, I think, in my mind is uh, I'd say unfortunate, right? That as a society, we don't really seem to be spending our resources in, areas that may matter.
Yeah, and I think that that there is a crossover between aging and chronic infectious disease, and it would be nice to see some, like, yeah, if you have something like HIV that harming a lot of people, you should put a proportionally high amount of resources into it. But then we should also be thinking, not putting zero resources into the more minor persistent infections that are also damaging us as we age, but, putting a proportional amount of resources into them. I think there could be a lot of great return on investment there.
Yeah, yeah. Yeah, maybe back to the house analogy, right? Like, I guess when people buy a house, they assume, well, every year you have to spend, X thousands of dollars on maintenance, right? And then, if the bank is giving you a mortgage for it or whatnot, they want to make sure that you can actually pay for anything that happens to the house, right? We don't go and say, well, yeah, let's assume that you need to spend, 1, 2, 10% of your salary on maintaining your body, right, over many years, right? Kind of sounds ludicrous, right? But that would be the equivalent, right? And it would be even more important than maintaining a house.
Of course. Yeah. 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 That's a perfect analogy.
Maybe kind of you know close up on all that, right? Like of course, apart from doing research on longevity and trying to develop longevity therapeutics, like what are some of the things that that you do yourself, or that you would uh uh that you would think that are good for people to do to keep themselves as young and well functioning as possible until we actually have therapies that can make an impact.
Well, everyone hates to hear this, but the most obvious low hanging fruit are, diet and exercise and sleep. You really want to nail those. You want to not smoke tobacco, and you don't want to consume significant amounts of alcohol, and that can all make a big difference in terms of rate of aging. And then you can get into the world of supplements and stuff like that, and I certainly take some number of supplements, but not dozens or hundreds the way some people do. I like to do a lot of research on things that I'm going to take, and I actually do a lot of testing on myself, looking at my biomarkers, looking at levels of vitamins and such like that. So I don't take vitamins. I don't take multivitamin with just like everything on the 100 things that they put in there. I only take the things that are actually low in blood tests that I get for them. Um, and then there's more biohackery things that you can get into. You can play with the aging clocks and then do little mini experiments on yourself and say, okay, if I start taking, this supplement or that supplement, does it reduce my epigenetic age? Whether or not, how well validated those things are now is definitely further away from the other things that I just mentioned. But it's stuff that you can play with. It's stuff that I play with a bit, and a lot of people that I know are playing with that kind of thing. And you can measure other biomarkers that are much more common sense, right? You can measure your blood sugar at home. It's very easy these days. You can measure your blood pressure, things like that. You have, your wearable devices that can measure your resting heart rate, your heart rate variability. I think heart rate variability is something that people could be doing a lot more self experimentation for, and something that I wish there was more attention from app developers, from machine learning and AI, trying to figure out what is it that we do that can raise our heart rate variability. So high look resting heart rate is generally good, and obviously you don't want to take it to zero or else your heart's not beating right. That's one of those things that kind of has a sweet spot. But generally the lower your resting heart rate is, the better, but the higher your heart rate variability, the better. And so it's something that I've played with a little bit, of trying to get my heart rate lower and my variability higher. I haven't had a lot of success at it yet, but it's something that's very easy to measure, and therefore self-experiment with and see what's working and what's not. And you can do it at population levels and, publish papers and put people on leads, but then you can do it on a very personalized level very easily these days by being able to measure it at home.
Yep, yep, yep. Um, the two markers that I remember hearing a while back and uh, that I like because they are probably arguably the easiest to measure grip strength and walking speed, right? 6 minute walking speed. That's right. Associated with longevity, right? You don't need absolutely anything to measure, any of those, right? Um, and it was actually you mentioned so smoking. I was talking with Christine Glorioso a while back, and then she mentioned also of course don't smoke, but the problem in smoking is not the nicotine, right? Like nicotine actually happens to be an interesting nootropic, I guess. The problem with smoking is all the toxins, right? In the kind of the burning of the tobacco, right? So now you're talking about thin pouches or whatever, and whether they're good or not good. Anyway, that was a fun conversation.
Yeah, yeah. I don't know much about that, but I have another friend in our community who's kind of going down that rabbit hole and experimenting with nicotine supplementation as I don't know whether it's a performance enhancing drug or whether it's has some sort of long term therapeutic benefits. But I don't know. I think it's probably worth staying away from getting addicted to something that is so easy to get by inhaling smoke on, and also just getting addicted to things. I just poured myself some more tea that has caffeine in it, so I've definitely voluntarily addicted myself to that. But I don't know. I try to not get addicted to too many substances. It just kind of complicates your life if you like have to remember to to get your fix on, this or that drug, and,
Mm.
this
I
is
think caffeine is probably better than sugar though. Like if you're going to get addicted to something, I would say caffeine is probably one of the better one.
yeah, and so I guess, the argument with nicotine, if you're taking it in a way that's not, not overtly damaging, like, inhaling smoke into your lungs, then, maybe it's, as harmless as caffeine. I don't know. But, yeah, I know it's something that people are interested in, and, I haven't yet, started experimenting with. I've managed to avoid nicotine my whole life, except for secondhand smoke,
Yeah, yeah.
growing up and all that.
No, I mean it's very funny. Right? Like I think we were talking about it because she mentioned Jean Clement, right? Like the oldest recorded woman, right? Like she took up smoking when on her 100th birthday or around her 100th birthday, something like that. And I was like, yeah, of course. I mean, if you have, I guess, for whatever reason, right? Like cancer, didn't really hit, right? And then I guess the main problem with tobacco is likelihood of developing like cancer, I guess, right? Like so if she for whatever reason was not likely to develop cancer, right? I guess the smoke of the tobacco was not necessarily as toxic for her or whatever. And she smoked, I guess, up until she died at 122, right? I think it was.
I think she actually I'm not sure I remember this correctly, but I wanna say that she quit smoking a couple of years before she died. But yeah, the whole paradox of, super centenarians, people who can live, significantly past, 100 years old is it doesn't seem to be super closely tied to lifestyle. Um, so,
It's definitely not, athletes and marathoners and like, super fit people, right? Like,
right, yeah, there doesn't seem to be any, enrichment for highly athletic people. But to your point before, there is some connection to, like, average, at least health span of having enough muscle on your frame. Once you get past a certain age, if you don't have enough muscle, then you can become very frail and less functional, and then be more likely to fall. And if you're more likely to fall, you're more likely to break your hip. And if you break your hip after a certain age, then you have like super high likelihood of dying in the next year, things like that.
Mhm.
But yeah, you're right. It is a paradox that, athletes don't seem to live past 100, a lot more than the rest of the population does.
Yeah. Yeah, I mean, I think there's still so much that we don't know, right? Like, there's, of course, a correlation, right, between, exercise and living longer, right? But beyond the certain point, I think that disappears, right? And I'm not sure anybody actually understands why, right? It's someone that is much more fit, right? Like, why doesn't that translate into, longer lifespan? I don't
Yeah, I mean, it seems in my reading of the data, it seems to affect average, lifespan more than maximum lifespan, and so your chances of living past 100 don't go up, but your chances of living, to, 80 or 90 go up by a lot if you're doing all the diet and exercise things right.
know. Yeah. We're already at the hour mark. I think time really, flew by. This is a very interesting conversation. Thank you so much. I don't know if there's anything else, that we didn't talk about, that you'd like to mention, or maybe like if somebody has questions or whatever. I guess they can go to Cyclarity TX.
Yeah, cyclaritytx.com is our website. You can also find us on LinkedIn and Facebook, and follow us and send your questions. And uh, it's uh, it's been amazing to talk to you, Javier. You ask great questions, and it's always fantastic to catch up. And it's been fun to talk about all kinds of different aspects of aging and medicine, and what we can do about them.
It was my pleasure. Thank you so much. Okay. It was a pleasure speaking with you as always. And yeah, hopefully, maybe after or whenever your drug goes to the next phase, right? And your phase one or whatever, right? I'd love to chat again and go a little bit deeper into it.
Yeah, yeah. Let's do it again, and I'll tell you when after our phase one is done. We're actually almost halfway through our phase one, and we'll have some data to present and to be talking about in a few months. So and we'll wrap it up this year. So let's talk again soon.
Awesome, exciting. Thanks so much.
Thanks, Javier.